Welcome back, everyone. I am Dixon Achimugumusa. In Module 1, we looked at the fascinating world of biochemistry. We explored the fundamental concepts of stereochemistry, the central role of carbon in biomolecules, and the intriguing phenomenon of chirality. Today, we will be embarking on a journey as we study the chemistry, structure, and functions of carbohydrates. Before we look at carbohydrates, let us review our understanding of stereochemistry. As we have learned, stereochemistry deals with the spatial arrangement of atoms in molecules and is crucial to understanding the structure and functions of biomolecules. Carbon, on the other hand, is the backbone of life. It serves as the foundation for countless biomolecules. Its unique properties, including the ability to form stable covalent bonds and a wide variety of molecular structures, make it indispensable in the world of biochemistry. Chirality plays a central role in biochemistry. We have explored how chirality arises from asymmetric carbon atoms and how it influences the properties and functions of biomolecules. You can find all this and find a link to Module 1 in the description below. Now let's apply these concepts to our study of carbohydrates. With our foundational knowledge in place, let's turn our attention to one of the most essential classes of biomolecules, carbohydrates. Join me as we unravel the chemistry, structure, and function of these fascinating molecules in Module 2 of our journey through biochemistry. At the end of Module 2, you will be expected to appreciate the concept of carbohydrates as a biomolecule as opposed to just as a class of food nutrient. Define monosaccharides and discuss the chemistry, structure, and functions of simple sugars. She will be able to relate stereochemistry taught in Module 1 to carbohydrates and be able to explain new stereochemical terms like enantiomers, diastereomers, epimers, and anomers. Define oligosaccharides and discuss the chemistry, structure, and functions of oligosaccharide. Lastly, you will be able to define polysaccharides and discuss the chemistry, structure, and functions of complex sugars. Carbohydrates are a group of naturally occurring carbonyl compounds, aldehydes or ketones, and they contain several hydroxyl groups. Even though carbohydrates are often associated with being a class of food nutrients, they are more than just a source of energy. They play crucial roles as biomolecules in various biological processes. Carbohydrates serve as essential structural components in living organisms. They act as a primary source of energy in many organisms. They participate in cell signaling and recognition processes. Certain carbohydrates play a role in lubrication and protection. Carbohydrates can also act as antigenic determinants, for example, the blood group antigens A, B, and O. Therefore, carbohydrates are an important biomolecule that must be studied for our understanding of biochemistry to be complete. Carbohydrates makes up over 50% of biochemical matter, and they are the most abundant organic molecules in nature. They are synthesized through photosynthesis by green plants, algae, and bacteria. Animals, on the other hand, rely on plants for carbohydrates. Carbohydrates are composed of carbon, hydrogen, and oxygen primarily. The carbohydrates serve many functions in the living organism. Some of their vital functions are metabolic and nutritional function. An example of this is that carbohydrates are the chief source of energy. The biological breakdown of carbohydrates supplies the principal part of the energy that every organism needs for various processes. They also serve as reserve or storage form of energy in plants and animals. Carbohydrate fiber in our meals helps to keep bowel function going smooth. Carbohydrates add on to the taste and appearance of food items. Carbohydrates also serve structural function. The insoluble carbohydrate polymers serve as structural and protective elements in the cell wall of certain microorganisms, exoskeleton of some insects, and in the plant cell wall. They form a major portion of the supporting tissue in the connective tissues of animals. Carbohydrates also serve for communication purposes, and some polymers of carbohydrates are of critical importance in intercellular communication in organisms. They are involved in cell recognition, contact inhibition, and also have antigenic properties of blood group. Some carbohydrate polymers lubricate skeletal joints and participate in recognition and adhesion between cells. Carbohydrates are also a source of carbon for biosynthesis of other compounds. Carbohydrates can be classified based on complexity, size, or functional group. According to complexity, carbohydrates can be classified into three main groups: the monosaccharides, oligosaccharides, and polysaccharides. The monosaccharides are the simplest sugars consisting of single polyhydroxyaldehyde or ketone group that cannot be hydrolyzed or broken down into smaller units under reasonable mild conditions. They serve as the building blocks for the more complex sugars. Oligosaccharides contain from 2 to 10 monosaccharide units joined through glycosidic linkage or bond. They can be broken down into constituent monosaccharide units. Polysaccharides, on the other hand, are polymers of monosaccharide units joined in a long linear or branch chain through glycosidic bond. Hydrolysis of polysaccharide yield many units of constituent monosaccharide. Polysaccharides serve several biological functions. On the basis of size, carbohydrate can be classified based on the number of carbon atoms in the structure. For those that have just 3 carbon atoms, they are known as triose, 4 carbon atom tetrose, and so on. And based on functional group, we have two types of carbohydrates, the aldoses and the ketoses. The aldoses have an aldehyde functional group, while the ketones have ketone functional group. Monosaccharides are also called simple sugars. They have an empirical formula of CH2O in brackets N, where N is equal to 3 or a larger number. Therefore, monosaccharide consists typically of 3 to 7 carbon atoms, and they can either be aldoses or ketoses. Glyceraldehyde is an aldotriose, while dihydroxyacetone is a ketotriose. As we have seen here, this is glyceraldehyde. This is an aldotriose. Triose because it has 3 carbons. Aldose because it has an aldehyde functional group. Whereas dihydroxyacetone is also triose because it has three carbon, and keto because it has a ketone functional group. Some of the general properties of monosaccharides are that one, monosaccharides are polyhydroxyaldehydes or ketones, and their derivatives, and they can have a potentially free aldehyde or ketone group. Monosaccharides are the simplest form of carbohydrates, and they cannot be hydrolyzed to other sugar units under reasonably mild chemical conditions. Generally, monosaccharides are white crystalline solids, insoluble in ether, sparingly soluble in alcohol, but readily soluble in water. Most of the monosaccharides have a sweet taste and char when heated. The monosaccharides with a potentially free aldehyde or ketone group are able to reduce metal ions under alkaline conditions. Hence, they are excellent reducing agents. Monosaccharides are amphoteric in nature. That is, they are capable of reacting as weak acids or weak bases with strong acids or alkali to form salt. All monosaccharides except dihydroxyacetone contain one or more asymmetric or chiral carbon atoms. And this brings us to the study of stereochemistry in carbohydrates. Stereoisomerism in carbohydrate chemistry refers to the phenomenon where two or more carbohydrate molecules have the same molecular formula and sequence of bonded atoms but differ in their spatial arrangement. These spatial differences arises due to the presence of chiral centers in the carbohydrate molecule. All the monosaccharides except dihydroxyacetone contain one or more asymmetric carbon atoms. If a molecule has more than one asymmetric carbon atom, it can exist in 2 raised to the power n stereoisomeric forms, where n represents the number of asymmetric carbon atoms present. Therefore, aldotrioses, aldotetroses, aldopentoses, and aldohexoses have 1, 2, 3, and 4 asymmetric chiral carbon and exist in 2, 4, 8, and 16 stereoisomeric forms. So you can see the glyceraldehyde has just one chiral carbon, and 2 raised to power 1 is equal to 2, giving us two stereoisomers of glyceraldehyde, the D and the L glyceraldehyde. Ketotriose, which is a dihydroxyacetone on the other hand, does not have any chiral carbon. So it can only exist in one form. 2 raised to power 0 is 1. For the ketotetrose that have just one chiral carbon, they can exist in two forms. The aldotetrose, on the other hand, are able to exist in four forms because they have two chiral carbon, and 2 raised to power 2 equals 4 based on the formula. Glyceraldehyde contains only one asymmetric carbon atom, as we have stated before, that is on carbon atom 2, and therefore can exist as two different stereoisomers, that is, as D and L glyceraldehyde. For sugars having two or more asymmetric carbon atoms, the convention has been adopted that the prefix D and L designate the absolute configuration of the asymmetric carbon farthest from the carbonyl carbon atom, or the penultimate carbon atom, or the last but one carbon atom, or the highest numbered carbon atom. The symbols D and L designate the absolute configuration of an isomer and not necessarily the sign of rotation of plane polarized light, as we will see later. The structure with the hydroxyl group on the right and the functional group on the top of the asymmetric carbon atom is designated as D. The structure with the hydroxyl group to the left and the functional group on the top is designated as L. So the hydroxyl group is on the left and the functional aldehyde group is at the top. So this is an L glyceraldehyde. Here we have the hydroxyl group by the right, the functional group at the top. This is D-glyceraldehyde. So in essence, every other structure that have the functional group at the top, the carbon that is farthest from the functional group and which is a chiral carbon, if the hydroxyl group is oriented to the right, such a carbohydrate is a D, and if it is oriented to the left, we have an L monosaccharide. All the monosaccharides except dihydroxyacetone contain one or more asymmetric carbon, and so they are optically active. Optical activities refers to the ability of a compound in a solution to rotate the plane polarization of plane polarized light when observed in a polarimeter. Optical activity is shown by all compounds capable of existing in two forms that are non-superimposable mirror images of each other. The optical activity is expressed quantitatively as a specific rotation. In this formula, alpha D25 is equal to observed rotation in degrees multiplied by 100 divided by optical path length multiplied by concentration in gram per 100 mil, where alpha is the specific rotation in degrees at temperature usually 25 degrees Celsius, and the wavelength of the light employed usually the D line of sodium, which is 589.3 nanometer. If the rotation of the beam of plane polarized light is clockwise to the right as the observer looks towards the light source, such a compound is designated as dextrorotatory with the sign small letter D or plus symbol. And if it is anti-clockwise to the left, such a compound is designated levorotatory with small letter L or the minus signs as symbol. So such compounds are called enantiomers of one another. So you then have the dextroenantiomer and the levoenantiomer. The dextrorotatory enantiomer will rotate plane polarized light clockwise, while the levorotatory enantiomer will rotate plane polarized light anti-clockwise or counterclockwise. For example, The specific rotation of D-glucose is plus 112.2 degree, which makes it dextrorotatory. And that of D-fructose is minus 93 degree, which makes it levo rotatory. So even though they are both D monosaccharides, glucose is dextrorotatory while fructose is levorotatory. So again, D and L does not indicate, the capital letter D and L does not indicate uh, the rotation of plane polarized lights. What indicates rotation of plane polarized lights are the symbols small letter D or L, or uh, positive and negative symbols. They, so it is the symbol plus and minus that refer to the direction of rotation of the beam of plane polarized light, but not the absolute configuration. The absolute configuration is determined by D and L stereoisomer. So the D and L stereoisomer of any given compound have identical physical properties and identical chemical reactivities, with two exceptions. They rotate the plane polarized light equally but in opposite directions. They react at different rates with reagents that are themselves asymmetric. Then one other very important thing is that the equimolar mixture of D and L stereoisomers is optically inactive, as the asymmetric carbon atom passes through a symmetrical intermediate during chemical reaction. Such an equimolar mixture of the D and L stereoisomer is known as the racemic mixture, or a racemic mixture or a racemate. And racemic mixture is not optically active because, just like we have stated before, the D and L rotate plane polarized light equally but in opposite directions. So if you have an equimolar concentration of both, they will cancel out each other. So we have mentioned enantiomer. We have other types of stereoisomers in monosaccharides. So carbohydrates exhibit various stereochemical features due to the presence of chiral centers, particularly in monosaccharides. The spatial arrangement of atoms around these chiral centers influences carbohydrate properties, including solubility, stability, and biological activity. Stereochemistry dictates carbohydrate conformation, affecting interaction with enzymes, receptors, and other biomolecules in biological systems. In linear chain structures of monosaccharides, we have three main types of isomerism: enantiomers, diastereomers, and epimers. Aldoses and ketoses of the L series are mirror images of their D counterpart. These two D and L forms of a sugar are known as enantiomers. L sugars are found in nature, but they are not so abundant as the D sugars. When two sugars have the same molecular formula but are not mirror images of each other, they are known as diastereomers. When two sugars differ only in the configuration around one specific carbon atom, they are called epimers. As we can see here, L-glucose and D-glucose are split mirror image of one another. We call such enantiomers. This kind of stereoisomerism is enantiomers. So L-glucose is an enantiomer of D-glucose. If you look at D-glucose and D-mannose, for instance, you can see that if you look at carbon 1, is the same. Carbon 2, the orientation around carbon 2 is different. Orientation around carbon 3 is the same. Orientation around carbon 4 is the same. Orientation around carbon 5 is the same. Orientation around carbon 6 is the same. So D-glucose and D-mannose only differ in the orientation around carbon 2. This makes them epimers. So remember, D-glucose is not a mirror image of D-mannose. Because they are not mirror image, but they differ only in the orientation around one chiral carbon. They are known as epimers. If you look at D-mannose and D-galactose, for instance, see they are the same around carbon 1. They have differences in orientation around carbon 2. They are the same around carbon 3. They have differences in orientation around carbon 4. The same on carbon 5, same carbon 6. So here we see that there's a difference between D-mannose and D-galactose around carbon 2 and carbon 4. And if the differences between two monosaccharides around their chiral carbon occurs in more than one chiral carbon, we call such a diastereomer. If it is only one, it is an epimer. If you compare D-glucose and D-galactose, for instance, you see that carbon 1 and carbon 1 is the same orientation. Carbon 2, same orientation. Carbon 3, same orientation. Carbon 4, different orientation. Carbon 5, same orientation. Carbon 6, same orientation. So they differ only around the orientation in carbon 4, which makes D-glucose an epimer of D-galactose. But mannose and galactose are diastereomers of one another. So in the linear chain form of monosaccharides, we have three types of isomerism: the enantiomers, the diastereomers, and the epimers. In cyclic carbohydrates, the main stereoisomers are anomers. Ring formation generates a new asymmetric carbon at carbon 1, also known as the anomeric center or anomeric carbon, thereby giving rise to diastereomers, which are labeled alpha and beta. These are diastereomers that differ in configuration at the anomeric carbon formed during ring closure in cyclic carbohydrates. The naming of anomers is based on the position of the hydroxyl group attached to the anomeric carbon in the D and L monosaccharides. The arrangement of the hydroxyl group attached to the anomeric carbon determines whether monosaccharide is an alpha or beta anomer. In the alpha anomer, the hydroxyl group on the anomeric carbon points in the same direction, that is the upward direction, as the ring plane. In the beta anomer, the hydroxyl group on the anomeric carbon points in the opposite direction, which is the downward of the ring plane. The Kiliani-Fisher cyanohydrin synthesis is a chemical reaction used to convert aldoses into their respective cyanohydrins. This synthesis extends the carbon chain length of carbohydrates and introduces a cyano-functional group, which serves as a versatile intermediate for further chemical modifications. This synthesis extends the carbon chain length of carbohydrates, and the synthesis proceeds through cyanohydrin aldonic acid lactone intermediates. The first step is to react the starting sugar with an aqueous cyanide, which undergoes nucleophilic addition to the carbonyl group of the sugar. The cyanohydrin resulting from this addition is heated in water, which hydrolyzes the cyanide into a carboxylic acid group that quickly reacts with itself to form a more stable lactone. There are two diastereomers of lactones in the reaction mixture that will be formed. They are separated and then reduced with sodium amalgam. The reduction steps involve the mild reduction of carboxylic acid functionality back to an aldehyde functionality. The newly formed aldehyde group is produced with two stereochemistry. So the product of a Killiani-Fischer synthesis is a mixture of two diastereomers called epimers. For instance, if you have the D-glyceraldehyde, after undergoing the synthesis, it produces two tetroses, D-erythrose and D-triose, which are epimers of one another. They differ only in the orientation around carbon 2. D-erythrose, upon the synthesis, will also produce aldopentoses, D-ribose and D-arabinose, and the difference is always around carbon 2, which makes them epimers of one another. In the same vein, D-triose will produce D-xylose and L-lysose. D-ribose leads to the production of D-allose and D-altrose upon synthesis. D-arabinose produces D-glucose and D-mannose. D-xylose produces D-glucose and D-idose. And D-lysose produces D-galactose and D-galose. An aldehyde or a ketone can react with an alcohol to yield a hemiacetal or hemiketal. Hemiacetals and hemiketals are compounds that are derived from aldehydes and ketones respectively. The same way aldehydes and ketones react with alcohol to form hemiacetals and hemiketals respectively, carbohydrates react intermolecularly to form rings. When forming a ring, 5 or 6 member ring is most favorable and will only be formed. The carbon 1 will be attacked by either the carbon 5 or carbon 6 hydroxy groups to form a 5 or 6 membered ring respectively. Up to this point, we have been representing monosaccharides in the straight form, linear form, called the Fischer projection formula. Fischer projection formula. However, in reality, the linear form is normally a minor, less than 0.1%. In the solid state and in solution, monosaccharides exist in cyclic hemiacetal form. Ring closure corresponding to reaction between the aldehyde group and either the carbon 4 or carbon 5. Cyclization involving carbon 4 results in 5-membered ring structured related to furan and therefore are designated as furanose. While hemiacetal formation with the oxygen on carbon 5 gives rise to an essentially strain-free and therefore more favored cis-membered ring, a derivative of the pyran. Hence, they are termed as pyranose. So your pyranose is a cis-membered ring, while the furanose is a five-membered ring. So the original facial projection formula. This is how the ring form can be represented in the linear form. But this is a better way of representing the ring or cyclic structure, and we call it the Howard projection or the Howard formula. And this, of course, is a Pyranus ring or a pyranoside. In aqueous solution, many monosaccharides act as if they have one more asymmetric center than is given by the open chain structural formula. Already we have discussed that because of the new anomeric center that arises. D-glucose may exist in two different isomeric forms, differing in specific rotation. The specific rotation for alpha D-glucose is plus 112.2 degrees, whereas the specific rotation for beta D-glucose is plus 18.7 degrees. These two sugars do not differ in elementary composition, but differ in physical and chemical properties. When the alpha and beta isomers of D-glucose are dissolved in water, the optical rotation of each gradually changes with time and approaches a final equilibrium value of plus 52.7 degrees. This change is called mutal rotation. It is due to the formation of an equilibrium mixture consisting of about one-third alpha D-glucose and two-thirds beta D-glucose at 20 degrees Celsius. The ring forms of the sugars are the ones that predominate in solution. Hazel sugars exhibit a diverse range of conformation. The Howard formula may be used to indicate the ring forms of monosaccharides, as we have said. The substituents on the carbon atoms are represented as extending above or below the plane of the ring. The lower half of the ring is thickened to indicate that it is the portion of the ring that is directed out of the plane of the paper towards the reader. You can see this portion is thickened compared to the portion that is assumed to be at the back of the plane of the paper. Sugar conformations are crucial for understanding their reactivity and interaction within biological systems. Sugar conformations can be described using various representations, such as Fisher projection, Howard projection. The most common conformations for pyranose sugars are their chair conformation. Conformation denotes the arrangement in space of atoms in a molecule, which can be achieved by rotation about single bond. Although the Howard formula gives a better indication of the true structures of sugar than do the straight chain forms, they do not represent the actual conformation. Hexoses and pentoses that have converted into pyranose or furanoses take on either chair, boats, or envelope conformation due to the tetrahedral geometry of their carbons. Pyranose rings can form either chair or boat conformation, while furanose sugars, which are less common than pyranose sugars, can adopt twist and envelope conformation. The twist conformation is similar to the chair conformation, while the envelope conformation has the ring oxygen in the plane of the ring. Although there are exceptions, most aldohexoses adopt the chair conformation that places the bulky hydroxymethyl group at the C5 terminus in the equatorial position. Reactions of monosaccharides. Understanding the reactions of monosaccharides helps with understanding how monosaccharides act in nature, and they have several reactions based on their carbonyl group. Monosaccharides react with dilute alkali to form salts known as carboxylate. This reaction is called aldol condensation reaction. Carbonyl compound can be reduced to alcohol through various methods. One method is the use of reducing agents like lithium aluminum hydride or sodium borohydride. Carbonyl compound can undergo reactions with other carbonyl reagents, such as organometallic compounds like Grignard reagents and organolithium reagents. This reaction can lead to the formation of new carbon-carbon bonds. Glycoside formation is a reaction specific to sugars. It involves the reaction of sugar molecule with an alcohol, resulting in the formation of a glycosidic bond. This reaction is important in the formation of disaccharides and other sugar derivatives. Sugars, especially reducing sugars like glucose and fructose, have the ability to act as reducing agents due to the presence of carbonyl group. They can undergo oxidation reactions, such as the reduction of copper 2 plus to copper 1 plus in the Benedict test. Acylation refers to the reaction of carbonyl compound with an acylating agent, resulting in the addition of an acyl group to the carbonyl carbon. Methylation involves the addition of a methyl group to a carbonyl compound. This reaction can be achieved using reagents like diazomethane or methyl iodide. Periodate oxidation is a reaction specific to carbohydrates. It involves the oxidation of vicinal diols, that is, two hydroxyl groups on adjacent carbons, in sugars using periodate ions. This reaction can be used to cleave glycosidic bonds and analyze protein structure. Oligosaccharides. Oligosaccharides are short chain monosaccharides, that is 2 to 10 units, formed through glycosidic bonds between monosaccharides. The glycosidic bond is formed most frequently between the anomeric carbon of one sugar residue and a hydroxyl group of another sugar residue. Structural diversity found in oligosaccharides are due to the presence of different monosaccharide linkages and anomeric configurations. Depending on the number of monosaccharide units that are linked, the oligosaccharides are further classified as disaccharide, trisaccharide, tetrasaccharides, and so on. Among these, disaccharides are the most important class because of their biological role and relative abundance in natural products. Oligosaccharides are important for cell recognition, immune response, and protein folding. Oligosaccharide synthesis occurs through a stepwise addition of monosaccharides, and oligosaccharide processing and modification occur in the Golgi apparatus. This is an example of an artificial oligosaccharide, sucralose. Disaccharides. These are a group of compounds. They are sugars composed of two monosaccharides linked by glycosidic bond with the elimination of molecule of water. So they generally have a formula of C12H22O11. Disaccharides are composed of two monosaccharides. Disaccharides can be classified as reducing or non-reducing sugars. Those with potentially free aldehyde or a ketone group can reduce fairly in solutions, hence are called reducing disaccharides. The reducing disaccharides have most of the properties of monosaccharide, that is, they can form oxazones and show mutar rotation. Disaccharides can be synthesized through condensation of monosaccharides. Disaccharides can be hydrolyzed to their constituent monosaccharide by enzymes such as maltase, lactase, and sucrase. Disaccharides are important for energy storage, digestion, and cellular processes. Disaccharides can be involved in various biological processes such as cell signaling and immune response. Some disaccharides may exist in white crystalline solids and are soluble in water and sweet in taste. One of which is sucrose. Sucrose is the most abundant oligosaccharide and is ubiquitous in plants. The production of sucrose in plants occurs through process of photosynthesis. It is generally manufactured from sugarcane and sugar beets. This is the Haworth's formula of sucrose. Sucrose is alpha D glucopyranosyl 1,2 beta D fructofuranoside. So the fructose is formed when an alpha D glucose and a beta D fructose are condensed, forming a glycosidic bond between carbon 1 of the glucose and carbon 2 of the fructose. Remember, carbon 1 of the glucose is the anomeric carbon, and carbon 2 of the fructose is the anomeric carbon. Therefore, they do not have a free aldehyde or ketone. So sucrose is not a reducing sugar. Sucrose is hydrolyzed by acid or enzyme sucrase or invertase into glucose and fructose. The specific rotation of sucrose is plus 65 degrees, And after hydrolysis, the specific rotation of the mixture is minus 19.84 degree. Such a change in the specific rotation from dextro to levo rotatory is called inversion, and hence the name inverse sugar. The reason for the inversion is that fructose is more strongly levorotatory than glucose is dextrorotatory. Maltose does not occur in nature, but it's only formed when starch is hydrolyzed by the enzyme diastase. Maltose is formed between two glucose molecules in alpha 1,4 glycosidic bond. Have an alpha D glucose that forms a glycosidic bond with a carbon 4 of another glucose, which gives us an alpha 1,4 glycosidic bond. And with this, it leaves this anomeric carbon free. Therefore, maltose is a reducing sugar because it has a free anomeric carbon, unlike sucrose that does not. It exhibits motor rotation since it can exist in both the alpha and beta forms. Of course, this anomeric carbon can exist in both the alpha and the beta form. Sprouting cereals have high content of amylase, which splits starch present to dextrins and maltose. Malt prepared from sprouting barley is an excellent source of maltose. Starches are also split to maltose by amylases present in human saliva and pancreatic secretions. Maltose is hydrolyzed into two units by the enzyme maltase in the intestinal juice. Lactose, also known as milk sugar, does not ferment as easily as glucose and hence is ideal as a constituent of milk of mammals. In the case of lactose, the constituents monosaccharides are galactose and glucose. Beta D galactose that condenses, forms a glycosidic bond with the carbon 4, the hydroxyl group on the carbon 4 of glucose, leaving a free anomeric carbon, meaning lactose is a reducing sugar. It reduces fairly in solution, but not Baford's reagents. And thus, that can be used to distinguish lactose from other reducing disaccharides. It should be noted that lactose is not produced in plants, and it is hydrolyzed by the enzyme lactase into its constituent hexoses, which are galactose and glucose. Isomaltose is a product of partial hydrolysis of amylopectin of starch and glycogen. It's made up of two glucose units joined through the alpha 1,6 linkage. This is one glucose. This is the anomeric carbon of the glucose linked to carbon 6 of another glucose. So giving rise to a 1,6 glycosidic bond and leaving a free aldehyde group, leaving a free anomeric carbon. So isomaltose is a reducing sugar. It is commonly used as sweetener in sugar-free products and is not fully absorbed in the human small intestine, making it suitable for low glycemic diets. Isomaltose is metabolized by the colonic microbiota, contributing to the production of short-chain fatty acid and promoting gut health. Cellobiose is a partial hydrolytic product of cellulose. It's made up of two glucose units joined by beta-1,4 linkage. Beta-1,4 linkage, two glucose units. This is the anomeric carbon 1 in a beta position linked to the carbon 4 of another, leaving a free anomeric carbon, which makes it reducing sugar. Cellulose is metabolized by some microorganisms such as yeast and bacteria to produce energy. Trehalose, also known as micose, is inhibited in the ability of plants and animals to withstand prolonged periods of desiccation, known as anhydrobiosis. Trehalose is made up of two glucose units joined in alpha 1,1 glycosidic bond. Alpha 1,1 glycosidic bond, meaning there is no free anomeric carbon and thus cannot be a reducing sugar. It is found in various organisms such as bacteria, fungi, and invertebrates, and is a major carbohydrate storage molecule in some organisms. Trehalose can be hydrolyzed to its constituent glucose molecules by the enzyme trehalase. Trehalose has potential applications in various fields such as food, pharmaceuticals, and cosmetic due to its unique properties of high water retention. It is involved in various biological processes such as energy metabolism, signaling, and immune response. Trisaccharides. Trisaccharides are oligosaccharides composed of three monosaccharides with two glycosidic bonds connecting them. Each glycosidic bond can be formed between any hydroxyl group on the component monosaccharide, resulting in trisaccharides that are diastereomers with different chemical and physical properties. Examples of trisaccharides include raffinose, also known as melitose. We also have melezitose, maltotreose, and kestose. Trisaccharides are found in various biological systems and have important roles in energy storage, cell recognition, and immune response. They can be synthesized through stepwise addition of monosaccharides and can be hydrolyzed to their constituent monosaccharides by enzymes such as trehalase and sucrase. Saccharides have potential application in various fields such as food, pharmaceutical, and cosmetic. They usually have low molecular weights and water solubility, making them suitable for use as emulsifiers, thickeners, and stabilizers in food and cosmetic industry. They have prebiotic properties, promoting the growth of beneficial gut bacteria and improving gut health. They have antioxidant and anti-inflammatory properties, making them potential candidates for the development of functional foods and nutraceuticals. They have ability to stabilize proteins and membranes under stress conditions, making them useful in the development of pharmaceutical and cosmetics. They have potential to modulate immune response and inflammation, making them useful in the development of immunomodulatory agents. The two examples shown here are non-reducing as they have no free anomeric carbon. So we have the raffinose and the melezitos. And all the anomeric carbons are used up. So they are non-reducing. Raffinose, stachyose, and verbascose are non-digestible short-chain oligosaccharides. They contain galactopyranose residues involving alpha-1 cis glycosidic bonds that human lacks the enzyme to hydrolyze. Of the tetrasaccharides, the important one is the stachys derived from raffinose. It was first isolated from rhizomes of Stachys tuberifera. Stachys consists of alpha galactose units and one glucose unit and one beta fructose unit, sequentially linked as galactose 1,6, galactose 1,6, glucose 1,2 fructose. It occurs during germination of seeds, together with related oligosaccharides such as raffinose. Stachys occurs naturally in numerous vegetables, e.g. green beans, soybeans, and other beans, and other plants. Stachys is less sweet than sucrose. It is mainly used as bulk sweetener or for its functional oligosaccharide properties. Stachys is not completely digestible by humans. Verbascose, on the other hand, is a pentasaccharide that is stachys, which has an additional unit of alpha D galactopyranose attached by a 1,6 glycosidic linkage to the terminal galactoside residue. Therefore, raffinose, stachys, and verbascose, which are all related, are non-digestible short-chain carbohydrates or oligosaccharides. Humans do not have the enzyme to digest them since they contain galactopyranose residues involved in alpha-1,6 glycosidic bond that humans lack the enzymes to hydrolyze. So they pass unchanged to the colon, where normal intestinal bacteria ferment them to gases, which can cause abdominal bloating. So sometimes when you eat beans, for some of you that complain of having bloating when you eat beans or some kinds of tubers, you can see what is responsible for them. The enzyme alpha galactosidase can break them down and thus prevent gas after eating beans. And this is available as an over-the-counter supplement. Polysaccharides. Polysaccharides, often referred to as glycans, are complex carbohydrates which are polymerized anhydrides of a large but undetermined number of simple sugars, which are joined by glycosidic bonds. Polysaccharides are long chain of carbohydrate molecules composed of multiple monosaccharides and all their derivatives. They are mostly insoluble in water, but form a sticky or gelatinous solution and are able to form hydrogen bonds. They are usually tasteless, apparently amorphous, and some are crystalline. They function for energy storage, structural support, and cellular communication. Upon hydrolysis by acid or enzymes, they are broken down into various intermediate products and finally into their constituent monosaccharides or their derivatives. They differ in the nature of their recurring monosaccharide units, in the length of their chains, and in the degree of branching. Examples include starch, glycogen, cellulose, chitin, and hyaluronic acid. Polysaccharides are abundant and diverse renewable sources of carbohydrates, and they have various applications in food, pharmaceutical, and industrial sectors. The natural sources of polysaccharides are plants, animals, microorganisms, and algae, and they produce different types of polysaccharides. Polysaccharides can be classified based on their function or their composition. Functionally, polysaccharides are either structural polysaccharides or storage polysaccharides. The structural polysaccharides serve as structural components of living organisms, e.g., cellulose in plant cell wall, chitin in exoskeleton of some insects, etc. As storage polysaccharides, they function as reserve or storage form of fuel in living organisms, e.g., starch in plants, glycogen in animal cells, etc. In terms of composition, polysaccharides can be classified into homopolysaccharides or heteropolysaccharides. The homopolysaccharides are made up of a single kind of monosaccharide residue, e.g., starch, glycogen, cellulose, chitin, inulin. The heteropolysaccharides, on the other hand, are made up of two or more different kinds of monosaccharide units or their derivatives, e.g., hyaluronic acid, heparin, pectins, gums, mucilages, chondroitins, etc. Cellulose is the most abundant organic compound of our planet, accounting for about 50% of all carbon. It is principal constituent of cell wall in higher plants, forming the main structural element. It is a linear homopolymer of glucose unit linked by beta-1,4 glycosidic bonds, linear unbranched polymer with high tensile strength, formation of intrachain hydrogen bonds between adjacent glucose units along the backbone, increasing the stiffness of the polymer chain, establishment of interchain hydrogen bond between cellulose chain creates a two-dimensional sheet rigid and organized structure with thermostability and resistance to environmental factors like humidity. Cellulose is insoluble in water and all organic solvents. It dissolves in concentrated sulfuric acid on diluting the solution and boiling. Glucose is formed as a final product. Partial hydrolysis of cellulose yields cellobiose, a disaccharide. Cellulase, an alpha glucosidase produced by many bacteria and fungi, hydrolyzes cellulose. The large amount of glucose present in cellulose is not available as a source of energy for humans due to the lack of enzymes capable of cleaving the beta-1,4 bond. However, ruminants can effectively use cellulose as they contain large bacterial population in the rumen capable of hydrolyzing it. Chitin. Chitin is also a structural homopolysaccharide and is found in the cell wall of fungi, the exoskeleton of arthropods, such as insects and crustaceans, and other organisms, such as earthworms and mollusks, providing them support and protection. It is made up of N-acetylglucosamine units linked by beta-1,4 glycosidic bonds, forming a linear unbranched polymer with high tensile strength. Despite its toughness, chitin is biodegradable and can be broken down by enzymes called chitinases, which are produced by various organisms including bacteria, fungi, and insects. It is the second most abundant organic substance on Earth and has potential applications in various fields, including biomedicine, agriculture, and environmental remediation. Chitin and its derivatives have potential medical applications, including wound healing, drug delivery systems, and tissue engineering. Chitin-based materials are biocompatible and can be tailored for specific biomedical purposes. Chitin is utilized in various industrial processes such as water treatment, food preservation, and cosmetics. Its properties makes it suitable for application ranging from filtration to thickening agents. Peptidoglycan. Peptidoglycan is a structural heteropolysaccharide present in the bacterial cell walls. The repeating unit of peptidoglycan is the muropeptide, which is a disaccharide composed of N-acetyl-D-glucosamine and N-acetylmuramic acid, joined by a beta-1,4 glycosidic bond. N-acetylmuramic acid consists of N-acetylglucosamine, which has a carbon-3 hydroxyl group, joined to the hydroxyl group of a lactic acid by an ether linkage. In the peptidoglycan, the carboxyl group of each lactic acid moiety is in turn linked to a pentapeptide composed of L-alanine, D-isoglutaminyl, L-lysyl, D-alanyl, D-alanine. The terminal D-alanine residue of the side chain of one polysaccharide chain is joined covalently with the peptide side chain of an adjacent polysaccharide chain, either directly as in E. coli or through a short connecting peptide, e.g., the pentaglycine in Staphylococcus aureus. In gram-negative bacteria, the third amino acid in the peptide stem is often a diaminopimelic acid, whereas in gram-positive bacteria, it is L-lysine. Peptide chain attached to the muramic acid can be cross-linked, forming a mesh-like network and provides rigidity and shape to bacterial cells. They are targets of antibiotics such as penicillin and cephalosporins. The peptidoglycan structure of bacterial cell wall is resistant to the action of peptide hydrolyzing enzymes, which do not attack peptides containing D amino acids. However, The enzyme lysozyme found in the tears and in the egg white hydrolyzes the beta-1,4-glycosidic bond of polysaccharide backbone of the peptidoglycan structure. Starch. Starch is a principal storage homopolysaccharide of the plant kingdom. It is made up of glucose as repeating units. It's a mixture of two components, amylose about 20% and 80% amylopectin. The amylose consists of long unbranched chains of D-glucose units, which are linked by alpha-1,4-glycosidic bond. Its molecular weight ranges from a few thousand of about 500,000. It gives blue color with iodine due to the iodine amylose complex, in which iodine molecule occupying a position in the interior of the helical coil. Amylopectin also has a backbone of alpha-1,4 linkage glucose units, but in addition, branch through alpha-1,6 linkages. The average length of branching is from 24 to 30 glucose residues. It gives purple color with iodine. Both amylose and amylopectin can be hydrolyzed by the enzyme alpha and beta amylase. Alpha amylase cleave alpha-1,4 linkages at random to give mixture of maltose and glucose units, while beta amylase present in plant remove maltose units successively from the non-reducing end. The intermediate product left after cleavage of starch by alpha and beta amylase is called limit dextrins. Neither of these enzymes can hydrolyze the alpha-1,6 linkage. Microbial glucoamylase can act on both alpha-1,4 and alpha-1,6 linkages of starch to yield glucose. Starch forms the major source of carbohydrates in the human diet and is of great economic importance. The important source of starch are seeds, fruits, tubers, bulbs, cereals, grains, and can also be found in protozoa, bacteria, and algae. Glycogen. Glycogen is the storage homopolysaccharides in animal. It's often called animal starch. It is present mainly in liver, skeletal muscles, and in smaller amounts in all other tissues. It is stored in liver and muscles of animals and split to glucose in the liver to maintain proper concentration of glucose in the blood to furnish energy. The amount of glycogen present in animal varies widely among the different tissues with diet and physiological state of the body. It is also abundant in mollusks, while glycogens like polysaccharides are found in some bacteria. Glycogen is a branch chain of D-glucose unit resembling amylopectin of starch. However, the branching through alpha-6 linkages is more extensive than amylopectin, with 8 to 10 glucose units between branching points. A glycogen molecule may contain as many as 30,000 glucose units. It is readily dispersed in water to form an opalescent solution which gives a reddish-brown color with iodine. It does not reduce Ferlin solution. Synthesis of glycogen occurs through addition of glucose unit by glycogen synthase. Branching is facilitated by glycogen branching enzyme. Breakdown occurred through glycogen phosphorylase and the branching enzyme. It is split to glucose in the liver to maintain proper concentration of glucose in the blood to furnish energy. Glycogen is regulated by insulin and glucagon. Insulin promotes glycogen synthesis, while glucagon promotes glycogen breakdown. Mutation affecting glycogen metabolism lead to glycogen storage disease like von Gich disease, Pompe disease, and McArdle disease. Destran. Destran is a unique member of the polysaccharide family. Formed by the condensation of glucose molecules, destran differs from the conventional glucan due to its branch structure. The main structure consists of robust alpha-1,6 glycosidic linkages between glucose monomers, with branches formed by alpha-1,3 linkages. These branching patterns distinguish destran from dextrin, another glucose polymer with simpler chain structure connected by alpha-1,4 or alpha-1,6 linkages. Dextran has versatile application in various fields. In medicine, it is a crucial component listed on the WHO model list of essential medicine. Dextran 70, a variant, acts as an anti-thrombotic agent by reducing blood viscosity and expanding volume in cases of hypovolemia. Surgeons use dextran to prevent vascular thrombosis by regulating platelet function and thrombus formation. There are several other polysaccharides that you can group them as homopolysaccharides, heteropolysaccharides, or as structural or storage polysaccharides, or as linear or branch chain polysaccharide. Just giving few examples, three examples of structural polysaccharides, three examples of storage polysaccharides, and giving a mix of homo and hetero polysaccharides. Now we'll be discussing carbohydrate protein interaction, which plays a crucial role in various biological processes, including cell adhesion, signaling, and immune response. Lectins are proteins that specifically bind to carbohydrates using their carbohydrate recognition domains. Lectins are involved in processes such as cell adhesion, signal transduction, and host pathogen recognition. This is a 3D representation of a lectin. Lectins and glycoproteins mediate specific recognition and binding events, while glycosaminoglycans and proteoglycans contribute to the structural integrity of the extracellular matrix and regulate cellular processes. Carbohydrate analysis techniques. Analyzing and understanding the structure and composition of carbohydrates is crucial for many scientific fields, such as biochemistry, nutrition, and pharmaceuticals. And there are several techniques. Monosaccharide analysis. Analyzing their composition is the first step in carbohydrate analysis. Techniques such as high performance liquid chromatography and gas chromatography can separate and quantify monosaccharide in a sample. These techniques often require derivatization to enhance their detectability. Another technique is glycosidic linkage analysis. Carbohydrates are connected through glycosidic linkages, which determine their structure and function. To analyze glycosidic linkages, techniques like enzymatic digestion, methylation analysis, and nuclear magnetic resonance spectroscopy are employed. Enzymatic digestion breaks down complex carbohydrates into smaller units for further analysis. Mass spectrometry. Mass spectrometry is a powerful technique used to analyze the molecular weights and structure of carbohydrates. Techniques such as matrix-assisted laser desorption ionization mass spectrometry (MALDMS) and electrospray ionization mass spectrometry provide valuable information about carbohydrate composition, branching patterns, and even stereochemistry. Structural analysis. Determining the precise structure of carbohydrates is essential for understanding their biological functions. Techniques like X-ray crystallography and nuclear magnetic resonance spectroscopy can provide detailed insight into the three-dimensional arrangement of carbohydrates. Glycan profiling. Glycan profiling involves analyzing the entire population of glycan present in a biological sample. Techniques such as lectin microarray, glycan microarray, and glycoproteomics are used to characterize and profile the glycan structures in complex biological systems. Bioinformatics and data analysis. As carbohydrate analysis generates large amounts of complex data, bioinformatic tools and data analysis methods play a crucial role. Tools for glycan structure prediction and glycomics database and statistical analysis software aid in organizing, interpreting, and correlating carbohydrate data. And this can be a future career goal for a biochemist to be a bioinformatician or a data analyst. This essentially brings us to the end of Module 2, which is the chemistry, structure, and functions of carbohydrates. These are books that you can read for more understanding of this topic, and I'll be available to take questions and observations from you in the comment section. Thank you very much.